Hello students, welcome again for a biology lecture. During last lecture, we have discussed about characteristics of genetic code, how these genetic codes are there and they are universal, they are in a triplet form, they are commonless, there is no overlapping, then the degeneracy and non-ambiguous. So various characteristics are there which are necessary to know about this because the protein before protein synthesis we should know all the details and rules of this genetic code. Now let us start with the new topic and before protein synthesis again these topics are important. Now here the first one that we are going to study which are helpful for to understand the further study and let us see about this in a PPT. The first one that is the mutation and genetic code. So mutation and genetic code we are going to discuss in a brief. So let us see here. So this is the mutation and genetic code. Let us discuss this in a short. The slideshow uh, from this mutation and genetic code. Here, the mutation word that denotes there is a sudden change in the DNA sequence. So, DNA sequence that means the nitrogen bases which are arranged in a triplet format. This sequence is changed, and due to the change in DNA sequence, there is a change in genotype. You know that genes which control and govern the characteristic or the characters from one generation to the next one. And when this DNA sequence change, then there is a change of genotype, and of course that affects to the characters. So the characters also change, and if the characters are changed, then this is the step further for the evolution. So evolution occur because of there is a change in the genes or the sequence of DNA, and hence this mutation is very important. Mutation takes place because of various factors. It may be physical factor, it may be chemical factor, it may be due to the environmental factors, and so on. But this change, that is a sudden change, that is most affected phenomenon in the life of living organism, and hence this sudden change, that is the mutation, takes place because of the DNA sequence. Now let us see what in details about this mutation, how the mutation, that means the change in the DNA sequence that occur. So this mutation may be occur due to the deletion. Deletion means what? If any one nitrogen basis or the sequence triplet codon that changes. So the total frame changes because of the deletion, and this is due to the shifting of some nitrogen bases or triplet codon, or even there is because of the deletion X plus and that causes the mutation. So in this way, the first point is that it is because of shifting or the deletion of nitrogen bases. Second thing is that it is the insertion duplication. Insertion or duplication. Insertion means what? There is a addition. There is a addition of uh, amino acid, or there is a duplication. That means this DNA that duplicate itself, and because of that there is a new sequence is added, and that is also cause of the mutation. So this is a deletion or insertion or the addition of new type, or in addition to that DNA sequence, it may cause to the mutation. Now one more thing is that there may be point mutation. So what is a point mutation? Point mutation is a change in the single base pair, change in the single base pair of DNA, and that causes the mutation, which is called as the point mutation. So best example of the point mutation is sickle cell anemia. So we have studied this sickle cell anemia in the first chapter. That is the uh, Mendel's law. In that, post Mendelian theories or neo Mendelism, you must know about this word, that there is a change in the structure of the RBCs, and this, instead of the spherical structure, these cells changes into the sickle like shape. Sickle like shape, that is a half moon shape, you can say it is a half moon shape, and because of that it loses the power to carry hemoglobin, and hence the oxygen cannot be carried towards the in the circulation throughout the body, and hence this is unable to carry oxygen, and that causes severe diseases in the human being, especially in human being, and in some cases that it may causes for the death. While when the hereditary character is there with the sickle cell anemia, it is called as the lethal gene, which causes the death of the baby before the birth, and hence this play an important role in the human body, and hence this is the point mutation. We can see that the single base pair may changes. So in this way, this is all about the mutation and the genetic code, how they interfere with one another. So mutation, it is a very important thing that we must study about the mutation, why this mutation occur, and what are the different effects of the mutation, why this mutation takes place, and what are the different causes or the influence of mutation, which leads for the evolution in some cases, that is the change in character that leads to the evolution. So this is all about the mutation and genetic code. Now, next thing that about the protein synthesis is role of tRNA. Role of tRNA, that is the transfer RNA. As the name indicates, it transfer. What it transfer? It transfer the amino acid. These amino acids are specific, and these specific amino acids are carried by the specific tRNA. So tRNA is also specific, which cannot carry any type of the amino acid, but it has to carry only the specific, a particular type of the amino acid, and this transfer T or tRNA. This is only for this transfer of amino acid from the cytoplasm, where the amino acids are present, and carry towards the site of protein synthesis. So this uh, amino acid, why it is need to carry this amino acid? Because itself amino acid cannot transfer and cannot recognize the site, cannot have such a type of the anticodons which are present on the tRNA. Especially if you see here, this tRNA, this tRNA act as an adapter molecule. So this amino acid does not have any capacity to read the codon, the codons and anticodons. So codons are present on the mRNA, and anticodons are present on the tRNA. So codons and anticodons are complementary to each other, and hence this tRNA is only the factor in between this mRNA and the amino acid, and hence it is called as the tRNA as an adapter molecule. So it play an important role for the protein synthesis. Now, this amino acid that attach to the tRNA towards the acceptor site at the free end, that is called as a three end. So that arm or you can see the strand of tRNA is free, and it is a 3 site. That means as the polarity, we have studied further that the strands have different polarity. So at the three end, which is called as a long arm of the tRNA. So tRNA, it is a clover leaf model or the hairpin model. Here we have discussing, uh, we are discussing about this tRNA, which is a clover leaf model. It has a uh, it has a three regions, or these are the three different arms you can see here. These are the three arms, while the uppermost region here, it is a free. You can see here, this is the arm or the upright arm, which is called as a three end, and the short arm is called as the five end. Okay, this three arm, it is having with CCA. This is the triplet codon, and this end is called as the amino acid attachment site, where amino acid which is activated by ATP, this attached to the three site. While this there are two arms, and the lower arm you can see here, it is called as the anticodon loop. Anticodon loop itself has at the base called as anticodon. So at this region, anticodons are present, which are complementary to that of the codons which are written on mRNA. So in this way, this tRNA has ability or capacity to recognize complementary codons. And as per the codon, anticodons are there. For example, see here, here C is there, cytosine, and the complementary codon are G, that is the guanine. Then second, it has the U, anticodon triplet in middle, while here on mRNA it is the adenine. And next is the G, that is guanine, and this is the cytosine. So on the mRNA, you can see here, these are the codons. Codons are given by uh, DNA, and they are transcribed to the mRNA. So as per this codon, anticodons are there, and tRNA has ability to recognize these codons. As per that, the complementary codons are there, known as anticodon, and they recognize the site and then get attached. So this site is which after the identification, it recognize and get fixed to here, and the upper portion of this three end, which is with amino acid attachment side. Now after the recognition of this codon and anticodon, that means from mRNA and the tRNA, it get attached, and the amino acid gets separated, detached from this, which helps for the protein synthesis. So in this way, this tRNA itself act as an adapter molecule, as the amino acid does not have any capacity to read the codon, and hence this tRNA is very important, which called as the adapter. So here, amino acid acceptor site, that is the three end, and for every amino acid, the specific tRNA is there. For every amino acid, specific tRNA is there. That means not any type of the amino acid get attached to the tRNA, but the specific amino acid get attached to the specific tRNA because of their identity, which consists of such a type of the ability to attach amino acid to the three side. So the specification is there. Specification is there, that is with the amino acid with the tRNA. Then uh, it is the initiator. tRNA is specific for methionine. Methionine, which is the denoted by AUG. So that is a initiator, means from this codon AUG, which is called as the initiation codon, and from this AUG, the protein synthesis process goes on. That means it start. It is also known as the initiation codon or starting codon. and it denotes a specific type of uh, the amino acid which is called as the methionine. So for eukaryotic it is methionine, and for prokaryotic this name is a different one, that is not a methionine but another one. So in this way there is a difference between this pro and the eukaryotic. Now this at the end of mRNA you can get here, that is a termination codon. So it is phi 3 direction. That means toward the phi end polarity, it is the initiation, and at the 3, it is the termination codon is there. And this termination codon starts with the UUG or UAG, like that. So it is a termination codon and it is a meaningless. So it is called as a senseless codon. It do not specify any type of the amino acid, but it only gives the information that to stop the protein synthesis process, and hence there is no any tRNA. There is no any tRNA transfer RNA for the stop codons. As this stop codon is there, UAG for example, then the protein synthesis process is stopped, and no such a type of tRNA is present, and hence this tRNA act as a adapter molecule. So this is very important thing that, so there may be question in the exam, why the tRNA is called as the adapter molecule. So we have to write all these points one by one, and how we have to prove that how it acts as an adapter molecule. So in this way, this is all about the tRNA as the adapter molecule. Now, let us start with the actual process of protein synthesis. So, protein synthesis, the process is called as the translation. Translation. So, first one, it is what? Transcription, and second is the translation process. So, let us see here and discuss about this translation process, where translation process is the actual process of protein synthesis, and as per the codons which are written on the mRNA. So as per the codons which are of course in the form of triplet, these codons of mRNA, and they are as per the written mRNA, these are translated, and with the amino acid they form the polypeptide chain. So amino acids join with this chain, polypeptide chain, and eventually it form the last, that is the protein. So protein is synthesized as per and totally governed by the DNA, which the codons or the message is given to the mRNA as per the need. This message is written. We can see that just like the architecture before the building of any structure. So there is a structure, or you can say the design that is made by the architecture or the engineer and engineer. So in this way, this is a design written on mRNA, and that is by DNA which type of protein is to be synthesized. It is totally governed and controlled by the DNA, and as per that, just like the blueprint, it is a mRNA, and this total formation of the protein or the synthesis takes place. Now, let us see what are the requirements for the protein synthesis one by one. So the first one, that is the amino acids. Amino acids are present in the cell, that is the cytoplasm of each living organism, and there are about 20 amino acids we know. So the uh, 20 amino acids are present in the cytoplasm. They are inside the protoplasm or the cytoplasm, and they are utilized as a raw material for protein synthesis. You know that what are the proteins? Proteins are the building blocks of the amino acids, and in this way, these amino acids are present inside the cell cytoplasm. Second thing is that what are the requirements? So amino acid as a raw material, they are attached, linked with the polypeptide and hence these are the amino acids. Then next is the mRNA. mRNA, what I told before, that it has the message and it is written by, or it is synthesized by DNA, and hence mRNA act as a message which type of protein is to be synthesized. It is written on the mRNA. Of course, this process, that is the transcription process, takes place inside the nucleoplasm, inside the nucleus, and DNA, as per the need, these two strands, they uh, are separated from each other. Unwinding process is there, and only that site or that point that is exposed, or you can say that is exposed, and this strand, as per the written on the DNA, mRNA synthesis takes place, that is the message is given. Then the second thing is that about the tRNA. Now, what is the role of tRNA? tRNA are specially for transfer of these amino acids. So, as I told before, that amino acids are present in the cytoplasm, but they should bring toward the site of protein synthesis, and that is done by the tRNA. So, tRNA act as an adapter, and activated amino acids are attached to this free end, that is a three end of the tRNA, and it joins to tRNA, and this tRNA that brings, just like the vehicle, just like the uh, lorry or the trucks which bring the raw material to the site of building, and hence such a type of the amino acids are reached, they are provided by the tRNA from cytoplasm to the site of protein synthesis. The next thing is that ribosomes. Now ribosomes are this next requirement, where which provide the binding site for this amino acids, and it joins these amino acids, they are joined with one another at the site, and this amino acid, then messenger RNA, are all totally, they are just bind with this ribosomes. So that means ribosomes provide the site for the building of this polypeptide chain. And of course, the next is the ATP, adenosine triphosphate, which are rich energy molecules. And during this ATP, those are utilized as a energy currency, and hence it provides the energy for the protein synthesis process. Then the magnesium ions, plus ions are required. These are mainly for uh, these ions. They play an important role because the two subunits of ribosomes, they get associated together due to the magnesium plus ions. And next one is the enzymes. So various types of the enzymes are required for the protein synthesis. We will study that different enzymes time to time when the actually we will start with the protein synthesis process. So the enzymes, they play an important role, and they should be there, must be there, and uh, in absence of enzymes, protein synthesis cannot take place. Then the last process, or the actually process of the protein synthesis, that is divided into three parts. First is the elongation, second is translocation, and release factors. So we will see that detail in the protein synthesis. So here, let us see about amino acids. What I told before, that those are 20 amino acids in the free state. That means they are in a naturally, they are present in the cell. Then the specific sequence of amino acids are there. That means in a 5-3 direction, the sequence of amino acid is very particular, and it is specific. If you change the sequence of amino acid, the proteins, they have again changed. That means they are particular. If the sequence change, they also changes. So the nature of protein synthesis, protein is by the particular or specific sequence of the amino acid. So the process of protein synthesis, this is in this central dogma, you can say here, it is by DNA, totally governed, controlled, and regulated. This is by the DNA and the messenger RNA. So RNA act as the link between the DNA and protein. So this is a important, just the mediator for the protein synthesis. So from DNA all message is given and protein is synthesized. Then the ribosome serves as a site. Ribosome serves as a site. As ribosomes, as you know, that there are two types, 70s and 80s type, where both the types they have two subunits. One is large, another is the small subunit. So during the protein synthesis only, these units they unite or join with one another, large and small subunits. So the line is there of the messenger RNA and large subunit towards the above and small subunit towards the below. So in this way, these two subunits join and they provide the site for the protein synthesis. So this is all about requirements and details of this. Now, next one it is the ribosome, which provides site, and these sites there are about four sites are there. The first site, that is one is the binding site for mRNA. So what I said before, that one site of the ribosome that provides binding for the mRNA, and there are three different binding sites for tRNA. So first it is for mRNA binding and remaining three. So total four are there. Three binding site for the tRNA. They are of different names. Those are known as the A site, P site, and E site. So A site, it is for the entry of this tRNA. Then it is moved toward the P site, peptidyl site, where this binding takes place. Actually, the amino acid bind with one another and form polypeptide, and then they push or moves towards the next. And this tRNA, which is now vacant, which gives amino acid. Now this tRNA become vacant, and this is moves or it push towards the E side for the exit. So in this way, such a type of different sites are present on the ribosome, and this ribosome play an important role for the binding of both. So message which is written on mRNA, so binding for that mRNA with the tRNA. So for tRNA, there are three sites known as the three binding sites, and that is A, P, and E. So in this way, this is all about the translation process, where different requirements are there and how they are available in the cell cytoplasm. What is the nature of this? All we have studied in short. Now the actual process of protein synthesis is there, and this protein synthesis we will study in the next lecture. So let us stop here, and remember that you must read again and again so that you will get clear cut idea about this protein synthesis. So protein synthesis is somewhat complicated to understand, but if you go in a systematic manner, this is very easier to understand and get over all the process of the protein synthesis. So let us stop here and we'll study this to the next lecture, that is the protein synthesis. Okay.